In this video, we will discuss about cell death by bacteria, bactericidal antibiotics, or in other terms, we can call it as how the antibiotic leads to uh, cell death that are infected. So, what happens when we take a antibiotic? These aminoglycosides interact with ribosome, and at the same time, this quinolone also interact with the topoisomerase, and uh, there is also disruption in the uh, we can say RNA polymerase that helps in the transcription. So here the thing that is described here is based on effect of aminoglycoside on ribosome and this quinolone. So what happens is that whenever this aminoglycoside interacts with ribosome, this NADH conversion to NAD plus increases, and because of this increase in the conversion of NADH to NAD plus, what happens is that there is a there is a formation of excess O2 minus, or we can say there is a this condition, situation, that is conversion of frequent conversion of NADH to NAD plus leads to hyperactivation of electron transport chain, and this will lead to excess formation of O2 minus. This condition is known as superoxide formation, and this superoxide formation will disrupt the FES cluster FEF cluster damages, and this leads to the open iron, or we can say Fe2 plus is prone to Fenton reaction, and the result of this Fenton reaction is that it leads to the formation of this hydroxy radical production and this OH formation. This will interact with the DNA, lipid, and protein, and interaction of this will occur in a way that it will triggers the cell death pathway for the infected cells, or the cells that are infected with this, that are interacted with these aminoglycosides. And there is also a parallel interaction that occurs when aminoglycoside interacts with ribosome, is that it leads to the activation of CPX and ARC, and this will also leads to the superoxide formation. This will also leads to this disulfide. This will lead to disulfide bond formation, and this promotes superoxide formation. And this thing is shown in the figure in the, or we can say panel in the right hand side. That is when the aminoglycosides, this it interacts with the ribosome. This leads to the mistranslation of protein, or we can say the misfolded protein formation occurs for the cell surface membrane proteins that are destined to cell surface membrane. And when the misfolded protein reaches the cell surface membrane, it will triggers this CPX and ARC-C pathway that will together cause envelope stress response, and this envelope stress response will disrupt this TCA cycle. That is the same cycle that is disrupted in the panel in the left hand side. So this disruption of TCA cycle uh, will leads to hydroxy radical formation, uh, and simultaneously it will also disrupt the cell membrane function, or cell membrane function in terms of maintaining the O2 and NO3 concentration within the cell or within the cytosols. So all this will promote this radical hydroxyl group formation, and this will triggers the superoxide formation and cell death. So, and these things, as we can say, these aminoglycosides function is happened on uh, this aminoglycoside works on two different pathway. First is that this hyperactivation of electron transport chain, and second one is at the cell surface membrane, that is activation of envelope stress response. And in this, the disruption by quinoline is not shown. So what happens in this case is that the quinoline interact with topoisomerase, so it will disrupt the supercoil formation of this DNA, and that's the and simultaneously also triggers the cell death pathway, uh, as it is shown in the small figure. However, the details is not shown in this figure. If you need to understand this thing in detail, that is how quinoline affects or triggers towards cell death pathway. You can go through the reference that has been provided in the description. Uh, as of as of now, or for this video, we can understand that this topoisomerase disrupts the supercoil formation of DNA, so that in somehow triggers the cell death response by the cells that are affected by this quinolone. And how this disulfide bond formation will lead to superoxide formation is still not known. Further studies are need to be done in order to understand how this disulfide bond formation leads to superoxide formation. So if you need to know more thing about how this antibiotic triggers cell death, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.